Okay, hi everyone. Please let me know if you can hear me, see my screen, and if everything is all right, because my house just got a power outage. So I'm not sure if my connection is good enough.
Yes, we
Also,
can hear you.
amazing. Okay, that's a pleasure to see you. Well, I mean, hear you all, and I hope you will be having a pleasure to hear me talk about my work. It's a great honor to present today at this conference. So thank you very much for organizers for organizing the conference and inviting me and making sure it is possible for me to present online. So this is my PhD work. My PhD is already done. I'm now a postdoc in Ottawa Research Institute. I'm continuing in a different field. So this is essentially a summary of my 5-year journey in the University of Waterloo under supervision of Zoe Leonenko. And as was just told, the work is again about lithium isotopes, effects in neuroscience. It's going to have a little bit of different focus as compared to Amina's work. Thank you very much, Amina. You just made my work very much easier because you gave the entire background that I was about to give. So first is why lithium? And lithium, as you already know, is a forefront of medication in the treatment of bipolar disorder. It was known for decades. And what is interesting is that we still don't know how it works, why it is so efficient, and what specific targets it is targeting. We also know that there are different drugs for treating bipolar disorder and like Parkinson disease or traumatic brain injury, but lithium carbonate stays the simplest, like it stands out as the simplest out of all of them, and also it's safe for kids. So this makes it even more interested as a drug. So this is to recap why are we interested in lithium. The next question is why lithium isotopes? So as you already know, natural lithium consists mainly of two isotopes, lithium-6 and 7, both of them stable. Chemically, they are almost identical. They have the same shape, the same charge, and they form the same type of ion in solution. They differ in two main ways, their mass and their nuclear spin, as you can see here. So in classical biochemistry, we would usually expect these isotope effects for such light elements to be same or almost the same or like with a slight difference because of the mass. But lithium isotope effects, like works on lithium isotope effect, had different scenarios. Some works reported large effects, some works reported small effects, and some works reported no effect whatsoever. So depending on the system depending on the systems that you study, you can encounter essentially any of these effects. And we're interested in why, and also we're interested in if we can find the specific target. So the next question is why quantum? So the reason this question overall, like about lithium and why is it quantum, what's the connection? This question becomes exciting because it's connected to the broader idea of quantum biology. So Matthew Fisher proposed that nuclear spins may play a role in brain through calcium phosphate clusters that are called Posner molecules or Posner clusters. In this hypothesis, phosphorus nuclear spins could act as long-lived quantum degrees of freedom. Lithium can enter the system and replaced the two lithium can replace one calcium in the middle of the Posner cluster. If it happens, then the question will be, will these Posner clusters with lithium-6 and lithium-7 inside, will they be different? Because we have different spins for lithium-6 and lithium-7. So this is just a hypothesis. And to be honest, in my work, we're not investigating it. It's just like something for the context for you to know that we are interested in this too. So the strongest motivation of my particular work was our own study of which we done in rat brain slices using the multi-electrode array, as shown here. So this work was led. Oh, I have electricity back, which is amazing. So maybe you will have an interruption of a signal because my internet gonna kick in any moment. Anyway, so in this work, we were measuring the EPSP signal that is coming from red slices, and you measure it by multi-electrode arrays. You can treat your slices, you can treat the slices with lithium-7, lithium-6, or just natural lithium, so almidons. We did these experiments with lithium chloride and lithium carbonates. And as you can see, as shown on this graph, EPSP signal increase if you treat the slices with lithium-6 and decrease if you treat it with lithium-7 or natural lithium. Since natural lithium is predominantly lithium-7, this is kind of expected. So this work was done when I just joined the lab, and I was thinking it's very interesting. We have to dig deeper, and find where exactly this effect happens. Because when we're talking about the EPSP or electrophysiology recording of the entire brain slice, there are so many things that are involved that you cannot pinpoint a specific target, like why this is happening or where this is happening. Could be anywhere. It could be on a cellular level. It could be on a cellular network level. It could be something to do with mitochondria. Maybe ion channels are involved. You never know. So my entire thesis work was focused on investigation or like on the search for the specific target that can lead to the effect we just saw when I joined the lab. The first step was to continue my work, and this is still ongoing. So here the idea is now, instead of taking the red hippocampal slices, you essentially grow the neuronal network on the electrodes. So it's going to be artificial neuronal network in terms of it's not taken from an animal, and you just repeat these experiments. This one is still ongoing, and this just to show the pretty picture of how it looks like. Maybe we will be able to find isotope sensitive behavior in a slightly simpler system, and this will tell us something. Maybe the organization is important, but it shouldn't be natural, or it could be natural. Or maybe being natural from like a living organism, is the most important part. So this is still ongoing, and this is one of the examples of the work that is already done. So one of the targets was the mitochondrial sodium, calcium, lithium exchanger, which is called in CLX. So mitochondria plays a central role in calcium handling, energy metabolism, and neuronal function. And CLX is especially relevant because it stands for the lithium exchanger. I think my presentation goes a little bit ahead of me. Okay. Yeah, CLX is especially relevant because it is specifically a specific exchanger for lithium exchange. It exchanges fluoresces outside the mitochondria, and then just injecting your solution with either lithium or sodium and observing the signal. So the results of these experiments are quite clear. We tried this with obviously just mitochondria to make sure there is no calcium signal coming out of mitochondria if nothing is added, with potassium to make sure that if you change osmotic pressure or just add any salt like potassium chloride, still nothing is happening. And then, of course, with lithium and sodium. And you can see that signal increases. This is just calcium fluorescence rate, essentially. So like a rate that happened in here at the first minute, you can see the increase in the signal, meaning that calcium is extruded from mitochondria. Essentially, this is like a baseline to verify that everything is working in our system. And then we tried it with natural lithium, lithium-6, and lithium-7, and you can see that there is no isotope effect on calcium efflux through CLX. Sorry. And the other thing that we tried is we tried to do mixtures of lithium and sodium. But again, we saw no difference in terms of differentiation between lithium isotopes. At least there is no differentiation for calcium flux. The second experiment that we did was ICP-MS, or essentially mass spectrometry analysis. So with this technique, you're able to measure lithium in buffer and lithium in mitochondria. So inside versus outside. The control experiment was, again, lithium versus sodium. And you can see that my presentation is misbehaving today. I'm very sorry about this. And you can see from this graph that if you treat mitochondria with equal concentrations of lithium and sodium, then in buffer the ratio stays 50-50, but in matrix it drastically drops, meaning that mitochondria really prefers sodium over lithium. Now, if you do the same with lithium-6 and lithium-7, again, in buffer it stays approximately equal or like 1-to-1. But once you consider the matrix, the ratio drops again, meaning that mitochondria prefers lithium-6 over lithium-7, and the difference is around 40%. You can see more columns here. If my presentation allows me, you can see more columns here. That's because this work was done with CGP and without CGP. CGP is a specific inhibitor for NCLX, which blocks its activity. And you can see that there is essentially, well, there is a difference between blocked and not blocked, but the direction of a change is the same. This is going to mean that the mitochondria somehow differentiates between lithium isotopes, but it's not due in CLX. It is something else in mitochondria that is able to differentiate between two different isotopes. Maybe it's mitochondrial membrane, maybe it's another channel, maybe exchanger, something. So that's another question to ask and another interesting direction to explore. So our next target was ion channels, because when you think about the PSP, of course, some ion channels are involved either before a PSP comes or during the PSP signal or after. A PSP is a signal that comes just before action potential, so you may assume that if there is a change in the PSP, there's going to be a change in action potential as well. So we were measuring potassium channels first because, yeah, the easiest one to work with, very well studied. We picked KCSA, which is potassium bacterial channel, and we decided to go with the most robust system possible with black lipid membrane. This system allows you to embed the potassium channel in the artificial lipid bilayer, and you can have a full control of what's inside your system, meaning whatever you measure, you actually measure the current from this potassium channel. Once inserted, you measure current. The system allows you to measure as small as 1 picoampere. And we did this experiment in two cases, in potassium solutions, because this is potassium channel, and in lithium solutions. And we found that there is no difference in conductance between potassium or lithium. So we also repeated these studies for lithium-6, but again, there were no difference between either potassium or lithium-7 or lithium-6. So essentially, potassium channel is not the target. Once this was certain, we decided to study another one. So another one is sodium channel. This time, we decided to go for mammalian sodium channels and using patch clamp electrophysiology. The reason for this is that sodium channels are way more difficult to overexpress, purify, and properly fold in any artificial settings. So you, technically speaking, or like in ideal world, you can do black lipid membrane on them, but it's going to require a lot of effort, and the success is almost impossible, I would say. So far, I saw maybe just one paper where people were able to do this, and they used a very sophisticated centrifugation protocol to essentially push the channels into the membrane. That's why, sorry again, that's why we decided to do the patch clamp technique, which is very robust, very well known, and the essence of it that you pick a cell, of interest, you patch it with electrode pipette, and you can measure the current that goes through the entire cell. We did a whole cell configuration, meaning you collect the current from all the channels that are activated in the cell. To make sure that you're collecting only sodium currents, you block all the other channels with appropriate blockers, chemicals, or you modify your internal and external solutions such so no other current is passing. You also have a specific protocol to follow, uh, for example, sodium channels are voltage activated, so you're measuring in any case you're making sure that you're measuring current only from sodium channels. As a result, you're getting an IV curve, current voltage dependence, and usually you can use several parameters to characterize this, which is we have where half of the channels are open, reversal potential, the coefficient of the slope, which tells you something about the behavior of this of these curves, and maximum conductance, which is here. So this is a flow experiment. Essentially, we patch a cell. We start with sodium chloride to make sure that we get in a signal, the cell is healthy, there are enough channels open, and then we go in sequence to lithium-6, 7, natural, potassium, cesium, and sodium. So all our experiments are essentially done on one cell to make sure that there is no differences in terms of how many channels we have per cell. This were done in different orders to make sure that there is no weird order-dependent effect. And the entire purpose of having potassium and cesium here is to just make sure that there is no current if there is no appropriate ions present in the solution. The last one just makes serves as a verification that the current is still here and the cell is still alive. So we did these experiments in SHS55 cells and iPSC-derived cortical neurons. The first ones are neuroblastoma cells that are very robust, very well studied, very easy to work with. And you can see immediately, even without me describing these graphs, because there are no stars and no specific lines, that there is no difference between lithium isotopes whatsoever in the cells. But once you go to the iPSC-derived cortical neurons, which are way more like it's a closer model to what we see in the rats, because these are just some cancer cells and these are actual cortical neurons, you start to see differences in which is a half maximum activation potential between lithium isotopes. The difference is still small, so probably it's not the hour target that triggers if a PSP result, but maybe also this difference add up to each other, and at the end of the day, you have a rolling ball effect. Okay, so this is it, and I'm going to go to results in future directions. So just a summary, my thesis asked whether biological systems can distinguish between lithium isotopes, and if yes, where and how the distinction may occur. So the answer is yes. In some systems, you can see the differences, but not universally. For example, there is no difference in lithium isotopes affecting calcium efflux or in CLX, but there is a difference in mitochondrial differentiation. There is no difference in lithium in lithium conductance in KCSA channel, or maybe in all potassium channels. But we also notice that there is no difference between sodium and lithium in the potassium channels, while it's well-known fact that there is a huge, twofold difference between sodium and lithium in bacterial channels. So while it's not very relevant to the mammalian organism, it would be very interesting to see if there is a difference in isotopes in bacterial channels. And if so, then why is it happening in bacterial but not in mammals? Maybe there will be some hints or some new ideas here. We also didn't see any difference in the mammalian channels if we're talking about SH55 cells, but we saw small difference in iPSCs. Again, very hard to interpret correctly here. Maybe there is a rolling ball effect, maybe no, and it's just like a subtle differences that are present here and there. One second. Yeah, so the other targets that still would be interesting to explore further going to be calcium channels, because they are related to Matthew Fisher theory, and also about they are related to calcium Hampton, which is very important for EPSP signal of action potential. Same idea for endoplasmic reticulum, and of course of cellular networks. Some of these works are still in progress. We have a wonderful team of masters and PhD students. I think the collaboration now grew up to 4 different labs across borders. So hopefully we'll be able to share way more results very soon. And yeah, I want to thank you for your attention, organizers for organizing the conference. My entire lab, you can see a lot of people listed here, and I think these are all people that helped me from different universities, labs, and departments. I also want to say that please join Learn and Collab. We do wonderful research, which is very interesting. You can scan the QR code to see our website. We are looking for new ideas and for more students. Thank you, and I'm happy to take any questions.